This video will cover the process of protein synthesis in eukaryotic cells, including transcription, translation, RNA splicing, and different protein structures. DNA transcription is a process by which mRNA is synthesized, and it occurs inside the nucleus of the cell. The first step in transcription is initiation, where RNA polymerase binds to a promoter region of the gene. This gene is non-coding, and it is located immediately before the gene's coding sequence. After RNA polymerase has bound to the promoter, elongation occurs. The template strand, or the antisense strand, is the strand being transcribed, running in the 3' to 5' direction, and the non-template strand, or the sense strand, is the one that does not get transcribed, running in the 5' to 3' direction. During elongation, the DNA strand unwinds, breaking the hydrogen bonds present between the complementary base pairs. DNA is read in the 3' to 5' direction, and mRNA is synthesized in the 5' to 3' direction, with RNA nucleotide being added one by one. In RNA, there are the same nucleotides as DNA, with the exception of thymine, which is instead uracil. The process continues to transcribe DNA until it reaches a termination sequence, which is also a non-coding sequence, located immediately after the gene's coding sequence. The finished product of transcription is pre-mRNA, which includes both coding and non-coding pieces. The next step is RNA splicing, or post-transcriptional modification, where non-coding sequences are removed. In this case, the non-coding sequence, or intron, GAG, is removed. Another intron, UGG, is also removed. The remaining exons are connected to each other, and a poly A tail is added to the 3' end, while a methyl cap is added at the 5' end. These are added to protect mRNA from RNase enzymes in the cytoplasm.
After mRNA exits the nucleus, it enters the cytoplasm and binds to the small ribosomal subunit of a ribosome, where translation will take place within the cytoplasm. A tRNA, which stands for transfer RNA, molecule carrying the amino acid methionine will bind to the parking site of the ribosome and to the start codon of the mRNA sequence. In order for a bond to form between the amino acid and tRNA, a tRNA activating enzyme is used. This enzyme binds to both adenosine triphosphate, ATP, and a specific amino acid. Aminoacyltransferase then binds this ATP molecule to a specific amino acid to form an amino acid AMP complex. This is linked by a high energy bond, as two phosphates are utilized in the process by releasing energy through phosphorylation. The correct tRNA molecule then binds to the active site on the enzyme, allowing for the amino acid to bind to the attachment site on the tRNA, and AMP is released in the process. As a result, the tRNA molecule is now charged and ready for use. The start codon in all mRNA molecules consists of the sequence AUG and codes for the amino acid methionine. In addition, all tRNA molecules contain a sequence of 3 nucleotides forming an anticodon, which is the complementary codon to the mRNA molecule. The first tRNA molecule will bind to the 5' end of the mRNA sequence with its complementary pair of codons that match its specific anticodon via hydrogen bonds.
The first tRNA molecule attaches to the parking site of the ribosome, and the second tRNA enters at the attachment site. Methionine detaches from its tRNA and forms a peptide bond with the second amino acid. When the ribosome moves down the mRNA by one codon in the 5' to 3' direction, the first tRNA moves to the exit site where it is released, and the second tRNA moves to the parking site with the 2 amino acids. A third tRNA now attaches to the mRNA at the attachment site. The amino acid chain transfers to the third tRNA and another peptide bond is formed. The ribosome continues to move down the mRNA and the fourth tRNA enters at the attachment site. A chain of amino acids, a polypeptide, forms. These steps repeat as a cycle until the ribosome reaches a stop codon towards the 3' end of the mRNA, which in this case is UAG. A release factor binds to the codon, signaling for translation to stop. All elements involved in the process disassemble. The polypeptide detaches from tRNA. The ribosome disassembles into its 2 subunits, and the mRNA is released. The resulting polypeptide is used to form protein structures. The polypeptide chain is the primary structure. It can form 2 types of secondary structures, alpha helixes and beta pleated sheets. The structures are determined by hydrogen bonds formed between non-adjacent amino acids. Secondary structures form the tertiary structure. Its three-dimensional shape is determined by interactions between the secondary structures, such as hydrogen bonds or disulfide bridges. The order of amino acids in the initial polypeptide chain influences the bonds that are formed, which determines the resulting shape of the protein. Lastly, a quaternary structure is formed by combining multiple tertiary structures, creating the final protein.